Good evening, everyone. Today's my code is P50 and my topic name is Key Challenges in Meeting Regulatory Requirements of Medical Devices. So, what are medical devices basically? So, medical devices are any instrument, equipment, or any apparatus which are used for the medical purpose, and it may be a pacemaker, it may be a bandage, anything. These are comes under the medical devices. So when we have to market these medical devices, so we have to follow the regulations, because regulations are very important and it play a crucial role in our pharmacy, because it gives a surety about our efficacy, safety, and the quality of our product, how it performs. So we get a clear idea about all these things of our device. And so regulatory compliance is very essential, as we all know, and there is not a common regulatory body for our medical device. According to the regions, it differs. Like US have the FDA, then our Europe have the EMA, Australia have the TGA. So it differs when the region changes. So if we have to market our product in US, then we should follow the guidelines according to the FDA. If we have to market our product in Europe, then we have to follow all the guidelines according to the EMA. Okay? So according to all the different authorities, we have to compliant with that specific regulations. So while meeting these such regulations, we faces a lot of problems. I mean, the manufacturing faces a lot of problems or challenges, because to meeting the regulatory compliance is not easier. So let's see what problems did the manufacturer faces during this compliance with the regulation. So first is our regulatory diversity across the region. Next is a complex documentation and testing requirement. Next is technological advancement, outpacing regulation, high cost and resource limitations, and frequent changes in regulatory standards. So let's see one by one. So first is our regulatory diversity. As I said previously, that when we vary the region or the countries, the regulatory body for each country is different. For example, US have FDA, Europe has EMA. So according to that specific regulations, we have to fulfill our compliance with them. And during this, the manufacturer have to completely make the documentation different for each and every country. For example, when we take about US FDA, then we have to give the 10K clearance. Then we have to submit the PMA, and if we see in European EMA, then we have to submit compulsory the PSM, means the post marketing surveillance, as well as the PMA. Okay, and for the classification class 3, we have to give the PMA, and it is essential because it has a high risk. So next is our complex documentation and testing requirements. So when we talk about the documentation, there is a number of documentation which we have to give to the authorities, and due to that it becomes time consuming, and it also requires a lot of resources. So it is kindly very this is a time consuming because each and every country has its regulation. So as per all the requirements of that regulation, we have to document it different, and we have to conduct the clinical trials. If they given some extra trial or extra clinical test, then we have to perform that also. Next is our technological outpacing regulation. So in this we can say that now our AI is very preferentially used in all over the sectors. So, for example, the tools, for example, Navina, then AI doc. So these are all the AI tools which are used as the medical devices now, and without knowing their guidelines or the what without knowing their about them, how to use them, it is difficult for the regulatory authority to give the regulations for it, and also for the manufacturer to market in the international market. Next is our high cost and resources limitation. So when we have to comply with all of these regulations, so it takes high cost and also time. And when we talk about the SMEs, what means the small and medium sized enterprises, then they face the problem of the cost, and as well as the resources, means the manpower, the financial, they face the financial problems during this all. Next is our frequent changes in a regulatory standard. One last. Frequent changes in a regulatory. So we know that when there is an update in a regulations. So, for example, the European EMA is recently updated in 2017. So, as per the update, we have to change our existing documentation process and everything. So, this is also challenging because we have to change our existing process as well as the designs. These are some solutions which we can prefer and try to prevent those challenges. So, we can collaborate with the other industry when we have to engage with the regulatory bodies, that we will have all the information before all that, and we should have the efficient document management system, as well as seek partnership which help for us to funding, and which will be cost effective strategies for compliance, and last to track the regulatory updates and adjust the compliance strategy accordingly. And conclusion, so we all know that regulation is very important when we consider the safety, efficacy and the performance of the device. So we have to be proactive and investigating inefficient system, collaborating with the expert and monitoring regulatory changes can help navigating these challenges, ensuring compliance support, patient safety, product effectiveness, and help manufacturer maintain a competitive.
Next participant, P16.
8 minutes. Good evening everyone, myself K K and my today's presentation code is P16. Today I am here to present my topic, requirements for clinical trials involving the medical devices. So the definition of medical devices is that medical devices are instruments, machines and other technologies used in pharmacies to diagnose and treat diseases. And they are also used in combination or with other. It is also the inevitably growing and becoming more important day by day. These are the topics which I am discussing today. This is the pie chart. It is shown that stents are used in 8.85%, the implants are used 8.20%, and the patient aids used as 13.20%. Consumables and disposables are used 16%, and equipments and instruments are highly used, that is 53.75%. Requirements for conducting clinical trials of medical devices. Guidelines of CDSO includes all information and reports should be in English only, and it is also expected that the information submitted, that is it is in hard copy or also it is submitted in the soft copy. Global perspective of medical devices. Distribution of medical devices trials in regions. This data is from 2015. Distribution of medical devices trials by region 2015. United States of Asia used 50% of medical devices. European market use 28% of medical devices, and other 22% are used by other regions. Difference between drug trial and medical trial in clinical trials. So in drug trial, number of subjects are used here about thousands, and in device trial only 100 to 200 devices are used. Randomization and binding. Drug trials are easy to perform, and in case of device trial it is difficult. Subject requirement. New drugs are studied carefully in phases, starting with introduction to a small number of healthy subjects. And in case of medical devices, specifically those drugs are required as surgical implants, because it is not possible to conduct the trials on unhealthy healthy subjects. Use of placebo. In drug trials there is possibility of use of placebo, and in device trials it is not possible. Phases of medical device trial. There are two major phases, that is, first one is pilot feasibility study, and second one is pivotal, that is confirmatory study. And there is also a third stage, that is post-surveillance study. Comparison between US and EU in medical device regulation. There is a difference in their regulatory bodies, that is US has the regulatory body, that is US FDA, and EU has the medical device directive body. Pre and post market supervision of medical devices. Within FDA, the Center for Devices and Radiological Health (CDRH) is primarily accountable, and the NDS are autonomous private enterprise to enact regulatory control. Risk classification. In US there are three tiered system classification, and in EU there is 4 tiered system classification. Conclusion. The regulation of medical devices around the world is very diverse. There has been an upsurge in the number, diversity and intricacy of medical devices. Medical device clinical trial plays a vital role in patient safety, efficacy. In India, medical device industry consisting of a wide spectrum of components and segments, which leads to a huge conduction of clinical trials. Thank you very much.
Next is P18, Sonali Holgi.
Today I am going to explain the requirements for clinical trials involving the medical devices. So introduction. So clinical trials are the primary methods which ensures the safety and effectiveness of the medical devices. It also helps us to build up the confidence among the patient, healthcare providers and among the regulators. Unlike the drugs, as we know, the medical devices are diverse in design and their functions. So there is difference between the clinical trials which are conducted for the pharmaceutical drugs and the medical devices. So this presentation covers the regulatory requirements, clinical trial phases and best practices for clinical trials. So as we know, there are 2 types of clinical trials, pre-market trials and post-market surveillance trials. First, pre-market trials are conducted before the device comes to the market, which ensures the safety and effectiveness of our product. Second is post-market surveillance, which is conducted after the product is available to the public use, which focuses on the long-term performance and safety monitoring. So these are four regulatory bodies which governs the clinical trials of medical devices. First is US FDA. The pre-market notification, that is 510K. In this process, where the manufacturer demonstrates that their device is substantially equivalent to the already marketed device. Pre-market approval, it is for the high risk devices which requires the extensive clinical data. Third is investigation, the investigational device exemptions, which allows us to use the device in clinical trials, which ensures the safety and effectiveness. Then EMA Europe, CE marking. CE marking allows us to sold our device in the European Union. This marking ensures that our device is compliant with the European safety, health and environmental standards. Then medical device regulation. These are the updated regulations in the market, European market. Then Health Canada, medical device license. Our device must be licensed before they are sold. Classifications. Our medical devices are majorly classified into the 4 classes based on their risk of level. Then fourth one is PMDA. PMDA is a pharmaceutical and medical device agency which is governing body in Japan, which is the overseas approval process for the medical devices. These are the phases of the clinical trials. As we know, there are 5 phases of clinical trials. Phase 0, which is an exploratory study where we understand how the device behaves in the human body. Phase 1, this is a small scale trials where we ensure the safety of our devices. Third is phase 2, where these are the larger scale trials where we assess the effectiveness of our devices. Then phase 3, where the large scale trials where we confirm the effectiveness of our devices. And last one is phase 4, which is a post-market surveillance where we monitor the performance of our device. Then designing of clinical trials. First is study design where we decide how to perform the clinical trials or how we design the clinical trials. There are 2 methods. First is randomized control trials and second one is observational study. After conducting the clinical trials, we are come to know the endpoint of our clinical study. That is primary endpoint and secondary endpoint. Primary endpoint mainly focuses on the safety and effectiveness, where secondary endpoint measures the quality and performance of our device. Then risk management. The trials must cover the ensures the patient safety and with the informed consent process. These are the then stakeholders in clinical trials, mainly regulatory bodies which gives the regulation to conduct the clinical trials. Then CROs, this is a third-party organization which recruit the patient requirement, then manage the data collection. Last. Then this, as my colleague already discussed, that challenges to conduct the clinical trials in medical devices, that is complexity of devices, recruitment, detection, regulatory delays and long-term monitoring. Conclusion and future trend. As we know, the clinical trials are crucial, so it ensures the safety and effectiveness. Regulatory framework provides the structure and ensures the effectiveness and long-term performance of our devices, and ongoing monitoring is necessary for every device. Thank you.
Good afternoon everyone. My topic is mainly requirements for clinical trials involving medical devices. There are the components which will be discussed. They are the we also know what is the medical devices. Already discussed after every conference. That will I skip it. Also the regulatory approval for the clinical trial. There are different agencies for the USA, the FDA, and for the global it is the ISA. So what is the purpose of the medical device trials? म्हणजे मेडिकल डिव्हाइस आपण त्याचं क्लिनिकल ट्रायल्स का करायचं? जर आपण जसं आपण मेडिकल याच्यामध्ये जर आपण एखादा टॅबलेट किंवा कशाचंही मॅन्युफॅक्चरिंग करतो, त्या प्रकारे आपण त्याचं प्री क्लिनिकल आणि क्लिनिकल ट्रायल्स करतो. त्या प्रकारे मेडिकल डिव्हाइसेसचं हे ट्रायल करणे इम्पॉर्टंट आहे. बिकॉज द मेडिकल ज्या प्रकारे आपण मॉडर्नाइज होतो किंवा आपलं AI आर्टिफिशियल इंटेलिजन्स जसे इंट्रोड्यूस होते, जसे आपली जनरेशन मॉडिफाय होते, त्याच प्रकारे आपल्या याच्यामध्ये जे इम्युनो सिस्टम आहे किंवा जी पण आपली रोगप्रतिकारक शक्ती आहे, अँड जे पण न्यू टाईपचे जे व्हायरसेस आहे, जे पण जसं पॅन्डेमिक आहे, असे वेगवेगळे व्हायरसेस हे येत आहेत. त्यामुळे काय होतं की जसं पण मॉडिफाय होतं त्या प्रकारेच पण डिसीज हे कसे होत आहे की ते वाढत जात आहे. त्याच्यासाठी फक्त आपण नुसतं मेडिसिन किंवा याच्यावर डिपेंडेंट राहू शकत नाही. त्यासाठी काय डिफरंट मेडिकल डिव्हायसेस हे मोस्टली प्रिपेअर केले जाते, ऑर्डर मॅन्युफॅक्चर केले जातात. ते डायरेक्टली कॉन्टॅक्टमध्ये येतात आपल्या जे पण आपण सर्जरीज, मोस्टली जे पण सर्जरीज असतात, फॉर एक्झाम्पल तुमचं युरिन टेस्टिंग ऑर द हिमोग्लोबिन, देन द फॉर एक्झाम्पल DNA अनालायझर, दिस इज अ डिफरंट काइंड ऑफ द थिंग. Risk management for the clinical trials. Mostly, clinical trials is important for they are directly involved in the our living system or the body. That's why the clinical trials is for the safety and the efficacy of the patient. It is important. Mostly काय होतं की जेव्हा आपण टॅबलेट्स आणि मेडिसिनचं जसं क्लिनिकल ट्रायल्स करतो त्या पण मेडिकल डिव्हायसेसचंही इम्पॉर्टन्स आहे, बिकॉज तो डायरेक्ट कॉन्टॅक्ट होतं आपल्या त्याच्यासोबत. डिफरंट मेडिकल ट्रायल्स इज इम्पॉर्टंट असतं, जसं तुमचं DNA अनालायझर आणि डिफरंट काइंड ऑफ थिंग्स. ती पुढे जाऊन एखादी सर्जरीमध्ये एखादा इश्यू क्रिएट होईल, कॉम्प्लिकेशन्स होईल, त्यासाठी डिफरंट ऑपरेशन्स असतात आणि प्रत्येक कंट्रीचे हे डिफरंट रेग्युलेशन्स असतात for the medical devices. Ethical standard. There are the preclinicals and clinicals trials for the patient. याच्यामध्ये इम्पॉर्टन्स आहे की तुम्ही पेशंटचे जे पण त्याच्यात तुम्ही करता त्यांचे कन्सर्न घेऊन त्यांचे इन्फॉर्मेशन घेऊन त्यानंतर अकॉर्डिंग टू विच डिव्हायसेस the sex, the age is matterable and आणि जी पेशंटची जी पण इन्फॉर्मेशन आहे ती कॉन्फिडेंटली असणं हे इम्पॉर्टंट आहे. डाटा इंटिग्रिटी इन क्लिनिकल ट्रायल्स. ॲक्युरेट डाटा कलेक्शन. जो पण डाटा आपण जो कलेक्ट केला आहे फॉर द क्लिनिकल आफ्टर द क्लिनिकल and pre-clinical trial, it must be done preserved or properly stored. Then the data management, proper storage and the security and organization of the trial data. Monitoring, जे पण तुम्ही आपण जे पण रेग्युलेटरी किंवा जे पण आपण क्लिनिकल ट्रायल्स केले त्याची मॉनिटरिंग करणे किंवा त्याच्यानंतर ते ट्रायल्स केल्याच्यानंतर त्याचं अप्रुव्हल घेणे, इट इज अ इम्पॉर्टंट अँड मॅन्डेटरी. Then importance. What is the importance for the medical trials in the clinical devices? Because the safety of the patient and the regulatory approval. जे पण clinical trials करतो in the medical devices, ते harmful patient मध्ये. That's why ते important आहे की तुम्ही approval घ्यावे जे standardize जे पण तुमचे bodies आहेत for the FDA, IOSA जे पण तुम्ही regulate करता त्याला fulfill करणे is important. Then the efficacy and the quality of the product is also the mandatory for it. Thank you.
And all. Under the category of research scholar, today I am going to present the oral presentation on topic formulation, development and evaluation of fast dissolving buccal films loaded with antiviral drugs. So the content of my presentation are introduction, aim and objective, methodology, research and discussion and bibliography. So fast dissolving films. The fast dissolving films are an innovative formulation technique. Mostly these techniques are used for increasing the customer acceptability because of rapid disintegration and convenience to handling, easy packaging, and also it has a pleasant taste. Fast dissolving film is a dosage form that employs a water dissolving polymer. There are number of advantages of the fast dissolving film that are such as these are suitable for pediatric as well as for geriatric patients. Then fast disintegration and rapid release should be achieved. Again, the onset of action is fast and then easily handled, and improved patient compliance should be achieved as bedridden patients or pediatric geriatric can happily administer it. They improved bioavailability, accurate dosing, and improved safety and efficacy, and again improved oral absorption. All these are the advantages of fast dissolving films. Then aim and objective of my study to formulate the fast dissolving buccal film of the anti-migraine drug. For my research proposal, I choose the lisdexamfetamine benzoate as an anti-migraine drug, and on that drug I will going to formulate I formulated the buccal film. So in this specially my objectives are to improve the its dissolution and disintegration time by using natural and synthetic polymers. Firstly, I am going to do the comparative study. Literature, the methodology, it includes literature survey, then selection of drug and excipients, then pre-formulation study, in that specially UV visible organoleptic properties, melting point, then UV spectroscopy, FTIR, solubility study, and comparison between compatibility study of drug and excipients to IR and DSC also. Then formulation of fast dissolving film, after that evaluation of that fast dissolving film, and optimization of batch from the trial batch, then comparative study with the marketed formulation, in vivo and ex vivo study of the fast dissolving film, and after that the solubility study of the fast dissolving buccal films. Then the composition of fast dissolving buccal films involve API, then film forming polymers which should provide the strengthening to the film, such as chitosan and guargum are the natural polymer, while HPMC is the synthetic one. I am going to choose the two grades of HPMC. One is having E3, which is having low viscosity, while one is E50, which is having higher viscosity. Then plasticizer, which will again provide the flexibility and improve the tensile strength of the film. Glycerin, sorbitol, mannitol are the examples of plasticizer. Surfactant and sodium starch glycolate and collapsible 407. These are the composition along with the details API, the concentration, standard concentration of API, which I told the lisdexamfetamine benzoate that drug should be taken 5 to 30, then film forming polymer 45 to 65, then plasticizer, surfactant, sweetening agent, saliva stimulating agent, as the drug is bitter in taste, and I am going to formulate the fast dissolving buccal film, so saliva stimulating agent is added to improve the dissolution and absorption. Elimination, coloring agent and flavoring agent should be added not more than 1% for. Then the trial batches, which now I am in the second year, so just enrolled in last year, so the trial batches were taken for the guargum, polyvinyl alcohol, methocel and hydroxypropyl methyl cellulose, out of which in that polyvinyl alcohol I face the coagulations, while in methocels there is somewhat bubble formation, so they both are rejected, and now I am going to finalize especially guargum and HPMC as a synthetic and guargum as a natural polymer. Then result and discussion, the fast dissolving buccal films were prepared by using the natural and synthetic polymer by solvent casting method. These are bibliography references.
Next part. You need a in vivo and ex vivo. What is the need to go?
Sir, as this is research scholar topic, so I need more justification or in depth. That's why I'm going to work on in vivo studies.
In vivo and ex vivo. why both are required or only one is sufficient? Otherwise, if you perform both, how will you correlate the two?
Mostly that is proposed.
So let's see one by one. So as we know that the oral route is most promising route for the drug delivery, and as it is a patient compliant route. So main challenge for oral administration that the drugs get degraded in the stomach. So the targeted drug delivery is not possible in case of oral route. So to prevent this challenge, we have to go for the gastric acid resistant drug delivery. Okay. So we use the natural polymer in this case. The natural polymer used for this drug delivery in my presentation, in my work, is nothing but a flaxseed polymer, that is flaxseed oxide. So as we know that the gastrointestinal tract are the dynamic nature. Okay. So mainly the gastrointestinal tract is divided into three main parts. First is nothing but the stomach, second is intestine, and the third is the large intestine, small intestine and large intestine. So the gastric acid residence time in the stomach is 2 to 4 hours, while the pH for the stomach is 1 to 3 generally, while for small intestine it is 6 to 8 and the residence time is 5 to 8. In large intestine is 5.7 to 6.7 and residence time is 24 to 72 hours according to the literature. Okay, it is by the literature only, not done any reference. So for preparation of the oral drug delivery or gastrointestinal drug delivery, we have to take into account that the drug should or the drug delivery agent should not dissolve in intestine for 2 to 4 hours and at the less than pH 3. So the main aim of the my presentation is gastric acid resistant drug delivery using natural seed isolate. As I already discussed that the natural seed isolate is nothing but the flaxseed. Okay. So first objective is to identify natural seed for formulation. Firstly, I have taken the 3 seeds, that is the flaxseed, then garden cress seed, and third is the tamarind seed polysaccharide. Okay. So I used flaxseed because the flaxseed have the according to literature, flaxseed have the capability to act as gastric acid resistant property. Okay. Then the next is the to isolate, evaluate and characterize acid resistant component from the seeds. The third objective is to formulate, evaluate and optimize gastric acid resistant dosage form, and the next is to generate and interpret the results. So the flaxseed is from family Liniaceae. It is also called as linseed. C is the word flaxseed. It is cultivated all over the world. So I use this methodology for my work. So first I have done literature review. I am in second year of the PhD. So literature review is for the 6 months. For 6 months I have done the literature review. Then after literature review, I have procured the flaxseed from the market and from the local market, that is RK, Nasik. After that, the authentication of seed is done from the pharmacy college only. After that, physiochemical characterization of seed and seed powder is done. The physiochemical characterization, that is the swelling index and loss on drying. Also the organoleptic properties and dissolution properties is evaluated in that case. Okay. Then after the physiochemical characterization, the pharmacokinetic characterization is done. In pharmacokinetic characterization, what we do that we react the seed powder with the Benedict's reagent. With the Benedict's reagent, the powder gives the blue color, which signifies that the seed contains the non-reducing sugars in large amount. That's what our aim is. Because the non-reducing sugar remain intact in the acidic environment. Okay. Then after that, the polymer is isolated from the flaxseed by taking the ratio of polymer, taking the ratio of the seed to the water in 1:10. And it is boiled for the 20 minutes. After 20 minutes, it is filtered through the muslin cloth. The mucilage will remain on the muslin cloth. The flaxseed residue will remain on the muslin cloth, and all the water residues will remain in the filtrate. After drying, we get the raw mucilage. Okay, so then we react this mucilage with the ethanol, and after reacting with ethanol, we get the white color precipitate, which is nothing but the mucilage of the plant. then we characterize the polymer similarly as swelling index, loss on drying, and other characterization acid resistance checked by the of the polymer. After that, we prepare the films. First, we prepare the films of the polymer by the solvent casting method. After preparation of film, we prepare the acid resistant capsule shell for the drug delivery. Acid resistant capsule shell is prepared with the help of the dipping method, dipping pin method. As we know that for preparation of capsule, for preparation of capsule shell, the steel material is used as a dipping pins. So we use that the dipping pins for the preparation of acid resistant capsule shell. We incorporate the drug into the capsule shell. After that incorporation, the evaluation was done. That is in vivo study and in vitro study was done. Only the part we have done that is up to the characterization of polymer till now. The other part is not done yet. Okay, next please. So as we know that the flaxseed have a significant swelling index, which show that the flaxseed has the property of water retention, and hence the ample amount of mucilage is present in the flaxseed. Okay, so the films prepared from the flaxseed mucilage were remain intact in the acidic environment. So the capsule shell were prepared from the flaxseed mucilage show the acid resistant property and show give the delivery in the intestinal region. Okay, thank you.
Myself, Mr. Jayesh, with my co-authors Survi and Shibra. We are here with a device named as wall-mounted incense emission analyzer. Our device is AI-powered with wireless connectivity and renewable energy sources. Our device is designed to detect the pollutants which are released into air due to burning of incense, such as particulate matter 2.5, then carbon monoxide, formaldehyde, and various types of volatile organic compounds. Our device generally identifies the pollutants in the air. It identifies the air quality and alerts us in the app which we have designed for our device. Over to Shibra.
We all are very much aware of the fact that incense burning is a part of our daily practice, which has some traditional, religious and spiritual significance in between the people, and it becomes very difficult to oppose a traditional practice or any cultural beliefs. And we also know that there are various products under the name of herbal products which are false, such as dhoops, agarbattis, which possess no natural essence but only synthetic chemicals which get combust and spread into environments by combustion. So hence, we today are here to address this very crucial issue. These are several factors which led to the making of the device. We all know that incense burning is a timeless practice. We cannot oppose a tradition, and we all know that according to modern sciences, any kind of smoke is harmful to our health, especially our respiratory health, because due to its fine particle size which penetrates the lungs tissue and may result in long-term lung diseases such as COPD, emphysema, and bronchitis inflammation. And recent detailed pooled analysis study showed that there is a 33% chances of getting a lung disorder due to the incense burning. And if we consume daily incense burning, like the smoke, it becomes 4 times more potent than one cigarette. So hence, this is a very crucial topic we are talking about. And this analyzer, which is our device, it is a best and good way to balance the traditional belief and our human health. Current methodology suggests that the traditional practice of incense burning is not properly monitored. Hence, people find it difficult to mitigate the potential health risk which the incense burning poses. And our device, which is the analyzer, it ensures and maintains the safety of air quality levels in our indoor spaces, such as homes and office spaces, or we can also apply it in temples where the levels of burnings are very high. Over to Sudhi. As the solution, we have proposed the solution to this problems is we have proposed a wall-mounted incense emission analyzers to design and monitor air quality and analyze emissions from incense burning in real times. We have used renewable energy sources such as solar panels or kinetic energy converters and to operate with sustainability. The AI-driven analysis ensures accurate readings and immediate alerts to users through wireless connectivity when emission leads to concerning. These are the features which are inbuilt: real-time analysis, sustainable energy source, discrete monitoring, wireless connectivity, and data logging. The wireless connectivity shows us that the device can transmit the data to the smartphone or computer via Bluetooth or Wi-Fi as per the user convenience. Over to you, Shipra.
So basically our analyzer uses different kinds of sensors such as temperature sensors and particulate sensors, which can you please hold on? So which analyzes the total information and converts or demonstrates into a real-time insights. This is a feature. You can see here we have demonstrated the graphs. So the indoor air quality. The sensor detects the toxicity level in the air and demonstrates into the graphs. For example, let's take example of carbon monoxide. So if the carbon monoxide levels are high, you can see here that above 1 the air quality is very poor and below 1 the air quality is good. That is, it is breathable in our indoor spaces. And based on this information, this is the feature called as real-time alerts, which is given to the users according to the toxicity level in our indoor spaces. For example, if the toxicity levels of carbon monoxide becomes high, it also suggests recommended actions to mitigate the potential health risk which it can have on human health. For example, if the carbon monoxide level exceeds, we should promote the ventilation in our homes or to check the source for its carbon monoxide. This is a prototype of our design for our real-time monitoring tool. Can you please open the simulator? So this is one of our feature. If the exposure level of a particulate matter is very high, suppose if it is for 52%, this is for the user convenience, which may have the health risk on the human system. For example, 66% there are symptoms including persistent headache, dizziness, respiratory distress, and for long-term health issues. So this is a simulator. Please open the model. For your easy understanding, this is the instrument. It is a 3D figure actually, can you project it? 230. So it looks like this, and for the user's convenience, this is the description of the instrument. This is the analyzer, components, and operation. Over to you, Dev.
So basically the working principle of our device is it has multiple sensors like VOC sensor, particulate matter sensor, temperature sensor, which collects the data regarding the air quality, which is displayed on onboard AI. It also logs the data for historical analysis of air quality, which helps and gives convenience to the user. Over to you, Surbhi.
We have much benefits of this app, of this product. Health monitoring, cultural recreation, then environmental appearances. It is user-friendly and data-driven insights. We have used these equipments for the analyzer, particulate matter sensor, VOC sensor, temperature sensor, solar panels, Bluetooth or Wi-Fi modules, AI processor, battery, user interface, data storage for the logging information. This is the different views of wall-mounted incense emission analyzer. Over to you, Shubham.
So we overall concluded that the analyzer is a best, we cannot say best, but a good way to maintain a balance between traditional beliefs, spiritual significance, and our human health. It also uses renewable sources of energy such as solar energy and which converts which will maintain the balance between traditional health and humans and
So basically what we conclude from our devices, it is we use solar technology, solar panels to improve sustainability. Then we have wireless connectivity which provides convenience for the use of users. Then we have AI use also, which makes it safer. It indicates us the quality of air, which
ensures and maintains the indoor air quality into safe ranges.
which will make us to change our daily routine. If we are using bad quality of incense sticks, dhoops, agarbattis, it will suggest us, it will alert us that change your products, shift to your products, etc.
Thank you.
Thank you. If any questions or your feedback.
So you have filed the design patent for it. What is the cost?
It will cost around 6,500 to 7,000.
You have prepared the prototype or
Yes, we have prepared. No, prepared. Prepared, sir.
Sir, Sir, we have created it basically for small indoor spaces such as homes, office spaces, or temples, where the incense burning optimize it. Yes, because people find it very difficult to mitigate its potential health risk, and it has a potential health risk on our respiratory system. Actually, there are some analyzers available in market, but they are all for outdoor air quality, which are basically used in various big industries. But there is no like for our homes and small spaces.
There are number of sensors which can be installed in our home, like VOC sensor, but they are all separate. We have combined each and every sensor in a device, which is also, we can say, pocket friendly. Even if it is of 6,500, but it is pocket friendly compared to other devices.
And all these things will be generated apart from the cells. Those will come from outside. So it will be quite difficult to manage our.
So we didn't also.
So this is about incense sticks, no? We are
Yes.
talking. But apart from incense sticks, there are several other factors.
Yes, sir.
We
So
can't control them, no?
yes, sir, we can control them.
We can analyze them.
Yes, we can.
We can't control them.
No, sir. We are analyzing the air quality. We
So
are not
basically our
controlling
objective
them.
is
We are
to analyze the
analyzing.
air quality and alert the user. Just like sugar machines, sir. शुगर कितना है वो डिटेक्ट करता है सर, बट शुगर को कंट्रोल कैसे करता है वो नहीं बोलता है। वैसे हमारा है कि हमको एयर की क्वालिटी बतानी है, कंट्रोल कैसे करनी है।
But the gases that are not monitored, oxygen, CO, otherwise nitrogen.
No, sir. Only pollutant.
Like formaldehyde, carbon monoxide, VOCs, particulate matter 2.5.
Ranges होते हैं, ranges, ranges, hazardous level,
hazardous levels are.
नाही तुम्ही कुठून आयडेंटिफाय केलं की PM 2.5 PM 10 याच्यावर गेला तर hazardous आहे.
So there is no rules.
So we have
So
also
how did you come to know that if PM 2.5 above this level is harmful to body or PM 10 PM particulate matter? Okay.
So we have done some actual research about particulate matters and we concluded the optimized ranges for it.
So you can go through EPA, US EPA, so you will get all these rules.
All our participants for your outstanding oral presentations. We appreciate the time, effort and dedication you put into preparing and delivering your talks. As we conclude our insightful oral presentations, we now move to the valedictory function of our one-day national level conference on regulatory aspects on medical devices in pharmaceuticals, sponsored by Dr. Babasaheb Ambedkar Technological University, Lonere, Caliber Biosciences Private Limited and Hexagon Nutrition Limited. It is with great honor that we invite chief guest for valedictory function, Mr. Raju Bhandari, Director of Caliber Biosciences, and Mr. Tushar Katkade, HR of Hexagon Nutrition, Nasik, and judges, Mr. Amol More, Head of Organization of Biotox Laboratories, and I now request to all chief guests and judges to come on stage. Sir, please. We extend our special gratitude to Mr. Bhandari and Mr. Katkade sir for their generous sponsorship, which has been instrumental in making this program success. Now I request Dr. D.D. Patil sir to felicitate Mr. Raju Bhandari sir. I request D.D. Patil sir to felicitate Mr. Tushar Katkade sir. Now I request Dr. R.D. Amrutkar sir to felicitate Mr. Mode sir. Now I request Mr. Raju Bhandari sir, Director of Caliber Biosciences, please interact with our students.
Good evening, respected dais and everyone present here. I have come to know that there is overwhelming response in this competition. It is a good sign. A pharmacist has a crucial role in pharmaceuticals and also in medical devices. Medical devices are tools to diagnose, treat and prevent illness and diseases. They can also be used to monitor, correct or modify treatment in patients. Medical devices used in body like pacemakers and outside the body like wearable sensors. Medical devices vigilance and post-market surveillance are important for the patients safety, product improvement, and regulatory compliances. These aspects have many steps in like classification, quality management, exhaustive clinical trials, and the healthcare and medical devices sector in India have significantly grown in the past decade. The wide range of medical devices from consumables to implantable devices are proudly produced in India. The Indian medical devices market share in the global market is estimated to be around 1.6%, and we hope to increase presence of this presence in global market. At the closing of this topic, everyone might think about opportunities in this field. Engineering, mechanical engineers, electrical engineers, biomedical engineers, and software engineers. The quality assurance, quality engineers and QC inspectors have a crucial role in this aspect. Of course, regulatory affairs. The regulatory affairs need managers, specialists to work on regulatory affairs, including US FDA submission, MDSAP audits. I thank management for inviting me as a chief guest. Please remember, excellence is doing ordinary things in an extraordinary way. Jai Hind, Jai Maharashtra.
Thank you so much, sir, for your fruitful interaction with our students. Now, before we announce the results, I would like to take some oral feedbacks from participants. So, please, anybody.
Attending this conference was a great learning experience. It provided clear insights into the regulatory requirements for the medical devices in the pharmaceutical industry. The speaker explained complex topic in a very clear manner to us, students like us, and it was very informative. The interactive discussion and the real-world examples helped in understanding the practical applications. Overall, it was a well-organized and insightful session. Looking forward to more such opportunities. Thank you.
The moment we have all been waiting for finally arrived. We are thrilled to announce the results of oral presentation competitions. First place is secured by Miss Shweta Pal. I request Bhandari sir to please. Second place is secured by Pranjal Thorat from Med Bhujbal College. I request Kakkade sir to give.
Sir, दोनों clear.
And third place is secured by Sankalp Zade from K K Wagh College of Pharmacy. I request More sir to give trophy and certificate. Congratulations to the winner for their outstanding achievements. Now I request R.D. Amrutkar sir, IPSC head and coordinator of second Pharmacon of K K Wagh College of Pharmacy to deliver vote of thanks. Okay, now we will have our consolidated prizes for this. Our first prize goes to Nusrat Sheikh. This presentation was conducted online. She might be absent here. Again, second prize goes to Aarti Shinde. This presentation was also conducted online. Then third goes to Saloni Gauri. Thank you, sir. Next prize goes to Ruchita Mahale. Thank you so much, sir. Here we conclude our prize distribution for today's oral presentations. Now I request Dr. R.D. Amrutkar, sir, to give a vote of thanks for today's conference.
Now it's good evening. Good evening to one and all. This thanks is such prayer that cannot be seen or touched, it must be felt by heart. I feel honored and privileged to get the opportunity to propose an vote of thanks of this second K K Wagh Pharmacon 2025. Firstly, I express my gratitude towards our beloved chairman, Honorable Samir Dada Wagh, for giving permission to organize this conference. I am very thankful to Honorable Professor K.S. Bandi sir, Secretary K K Wagh Education Societies, and Dr. N.S. Patil sir, Coordinator Pharmacy Institutes K K Wagh Education Society, for their constant guidance and the support. I am also thankful to respected Doshi sir, Finance Manager of this K K Wagh Education Society, for financial support for organizing this conference. Today my words are not enough to express the gratitude towards Mr. Bhandari sir, Director Caliber Biosciences Private Limited Nasik, Mr. Kelkar sir and Katkade sir, Hexagon Nutrition Nasik, for providing sponsorship for the conference. I also express my gratitude towards the Dr. Babasaheb Ambedkar Technological University Lonere for providing the assistance to this conference. My special thanks to Dr. V.K. Moria, who graced us with the kind words during this inauguration. My sincere thanks to all distinguished invited speakers for accepting our invitation and delivered useful talk of their expertise. Further, I am very thankful to all the judges, Mr. Amit Patil sir, Dr. Dake sir, Dr. Amol More sir, for accepting our invitation and providing inputs on this oral presentation. On behalf of this organizing team, deep regards and hearty thanks to Honorable Principal Dr. K. Saranki sir for their support in all possible manner to organizing this event. I would like to thank all the delegates who bless us with their presence. I am very thankful to all my faculty colleagues and non-teaching staff members who always stand by us. I feel proud that thank you for making this event successful one. An event of this dimension cannot happen overnight. The wheels start rolling days in advance. We have been fortunate enough to back by a team of very motivated and dedicated students, committee members and the volunteers. I cannot thank everyone enough for the involvement they have shown and the willingness they have expressed to take on the completion of the task beyond their comfort zones. I would also like to thanks to our photography and catering team. Without this, the event was not possible. Thank you very much again. At the end, I would like to say if time is money, then today we have spent millions of for us. Thank to all of you for making this event successful. Thank you. Thank you.
Thank you so much for your heartfelt words, sir. At the end, I conclude our one-day national level conference on regulatory aspects on medical devices in pharmaceuticals, sponsored by Dr. Babasaheb Ambedkar Technological University, Lonere, Caliber Biosciences Private Limited, and Hexagon Nutrition Limited. Good evening to all. Thank you.